Before we get into talking about actual experimental designs, I want to spend this week just reviewing the topic of sampling. Well, what do we mean by sampling? Well, sampling refers to selection of some part of a population to estimate something about the whole population. So, for example, if we were conducting an experiment on the Pawnee National Grasslands on the effects of grazing on grass biomass, we wouldn't go out and actually measure the biomass of all the grass on every plot on our study area. We would sample some small portion of each one of those plots or pastures. If we're looking at how thinning of ponderosa pine forest might influence the number of birds that occur, say, in an area in Klamath National Forest, again, generally we wouldn't try to sample the entire area that has been thinned. We're going to go in or get information on the entire area. We would go in and select a sample, and then we would make inference to that area that we're looking at. Another area that we're all familiar with where we've seen the results of sampling is, for instance, in presidential polls, where various polling groups will go out and call people and get an idea of who they're voting for, and then make predictions about who's going to win a particular race. Just to complete this idea of sampling, I also want to mention census, which is a complete enumeration of a population. And generally, we don't do censuses simply because of time and logistical constraints. Well, why do we sample? Well, as we talked about previously, it may be physically prohibitive. So in the case of the pasture, where we had different levels of grazing, we want to look at grass biomass, collecting the grass that's been produced over a year and across a entire, say, 2-hectare pasture may simply be impossible. It's also fiscally prohibitive, right? So it's going to cost a lot if you're trying to census an entire area. If you can get a good estimate with a sample, it makes a lot more sense. Even if a census can be taken, sampling is often cheaper. So even in laboratory studies, for example, where you're looking at petri dishes as your plots, in a sense, and looking at the effects of antibiotics on growth of some bacteria, rather than censusing the entire petri dish, which is probably possible, it's going to be a lot cheaper to just sample small portions of that petri dish and make an inference to the total population. Timeliness. If you want to get your results out in a timely fashion, which we all do, sampling is going to be a much more efficient way. And then finally, in many of the types of sampling that we do, it's destructive sampling. So if we're catching fish in a gill net, like in the picture here, virtually all or most of those fish are going to die in the process of trying to get this estimate of abundance or size or whatever we're interested in. And we obviously don't want to destroy the entire population in order to answer our question. So taking a small sample or a sample that's sufficiently large to answer our question is going to be a much more ethical way to conduct our experiment. Before we go on here, I want to make a distinction between sampling design and experimental design. In experimental design, we deliberately perturb or add treatments to some part of the population to observe what the impact is. And most of the focus of experimental design is what is the most efficient way to do that? Do we just simply randomly apply the treatments? Do we use what we call a blocked design, where we put all of the different treatments in one blocked area and then have that repeated across our landscape and so on? And we'll be getting into that in more depth. Sampling design, on the other hand, refers to how you estimate a quantity without necessarily perturbing it or having treatments. So you might just be interested in what is the average size of trees in a national forest. You're not going out and doing any treatments. You're not perturbing anything.
You just want to know what is the basal area of Douglas fir that we have on this national forest, or what is the average productivity of grass on this grassland, or um, number of fish per mile in streams in this particular area. So you're not necessarily testing a hypothesis, you're simply estimating some quantity. Now, sampling can be important in experimental design if you decide that you are going to sample some portion of each one of your treatment plots, for instance, rather than try to collect information across the entire treatment area. And we'll talk about examples of that. So knowing the basics of sampling design is very important to conducting good experiments. So just a quick review, why are we interested in sampling? As I mentioned, because we often sample our plots when we're estimating the response, even in a truly manipulative experimental study. Also, we need to sample when we use forecasting. So if we're trying to estimate the amount of wheat that's being produced across the U.S., we will go out and take samples across the area. We have a model that incorporates maybe precipitation, temperature, soils, and so on. And then we forecast there's going to be so many billions of bushels of wheat produced this year. And as I, again, also mentioned, the inference we make from an experimental treatment will depend on the samples that represent the population. So just a simple example here, if you do a thinning experiment in a forest and you just do it in one small district of that national forest, for instance, making inference to the entire national forest or across a larger area can be problematic, and it's at least important to be aware of that limitation. So now I want to talk about three critical terms that we're going to be talking about and using throughout the course. So it's really important to get these down. The first is what we call target population. Sometimes you'll hear statisticians using the term sample frame. We're talking about the same thing. It's a collection of all sampling or experimental units about which one would like to make an inference. And when we say an inference, we would like to take our sample and be able to say that this is a representative sample of that population or sample frame. So for instance, if we're interested in blood cholesterol levels of bears in Colorado, our target population would be all black bears in Colorado. And you think about that, that's actually pretty difficult to really be able to say that you are able to sample any individual bear in Colorado. The important thing for the target population is that every individual in that population has the possibility of being sampled, and we'll come back to this. If you're in fisheries, it might be halibut in Prince William Sound. For work that I do with the Bird Conservancy, we have divided up a number of western states, for instance Wyoming, into 1 square kilometer grid cells, and we randomly choose from those grid cells to go out and do our surveys so that we can make an inference to bird populations across the entire state. Sample unit refers to one of the units into which a target population or sample frame is divided for the purpose of sampling. In many cases, that's very clear: individual bear, individual halibut, 1 square kilometer cell. Although when you start thinking about it a little more carefully, you might want to define those a little more precisely. So you may be only interested in bears that are over 1 year old. Okay, so if you catch a cub, you may not want to use that in your sample. Same thing goes for halibut. If you're looking at some length-weight relationship or levels of
mercury or some other environmental pollutant in the tissue, you may again want to sample a certain age class or a certain size and so on. 1 square kilometer grid cells, that's pretty straightforward. Generally, you would want to make, again, everyone available for sampling. In many cases, there may be limitations to your sampling effort that would say, well, we can only really sample cells that are within a mile of a road or something. And then the sample refers to the set of sample units that are selected from a target population or sampling frame using a defined procedure. And that has been bolded because that's a really critical part of this. So if you just go out and select sample units willy-nilly and try to publish something on it, you're probably going to get hammered by your reviewers, because you need to be very clear about exactly how those sample units were selected. And that's really kind of the meat of sampling design, is how do we choose those samples such that it's representative, covering all of the different sources of variation that we're interested in, and so on. So in our examples, it might be that you have a budget to sample 15 bears or 120 halibut or 31 square kilometer grids. That collection of sample units would then be your sample.
A key concept related to sampling is what we call external validity. External validity is the extent to which the results of a study can be generalized to the world at large. And this is obviously key to doing science, right? We want to do something that doesn't just apply to the particular situation that we looked at. We want to be able to extend, generalize our results to the population of entire state, entire country, what have you. The degree to which a study has external validity depends on having a valid sampling protocol. And in order to do that, you have to do two things. First, you have to precisely identify the target population. Is it all of the squirrels in Arapahoe National Forest? Is it all of the squirrels in Colorado? Is it all of the red squirrels in the western United States? Obviously, the definition of the target population will determine the area, in this case, over which you're sampling. The second thing that's key is to use a selection protocol that ensures all sites, individuals, experimental units, whatever you're sampling, have a non-zero probability of being selected. So in order to generalize from your sample to a larger population, you have to ensure that every item within that larger population at least has some probability of being sampled. And we know that for some surveys, for instance, a breeding bird survey, we know that simply is not the case. The breeding bird survey was designed to be done by volunteers along secondary roads, and so all of the surveys are done right at the roadside. And many people have criticized the breeding bird survey as a result, saying, well, you really can't make an inference to the entire western United States or the United States as a whole, for instance, because you haven't sampled areas that are farther away from roads or that are close to large highways or in urban areas. So it's important to know exactly what your area of inference is, and we'll come back to this, but just really want to emphasize this idea of external validity. So let's start with random sampling, or sometimes referred to as simple random sampling, SRS. Simple random sampling is the best way to sample a population if you have no other information. It ensures valid statistical inference. And the basic property of a simple random sample is that every plot or every individual in the population has an equal probability of being chosen within your sample. So in this case, where we have a 20 by 20 grid, and we've got a sample size of 10, then each one of those cells within that grid has a 1/20 or a 5% chance of being included in our sample. Now, one thing that you have to be careful about is you can't tell whether a sample is random simply by looking at it. Okay, so these 10 points that you see here were chosen randomly, and you see that they're not well distributed across the entire study area. That upper right hand corner is not well represented by this particular random sample, and you will often see that kind of thing occurring. Doesn't mean that this is an invalid sample. It simply means that there's variation from one random sample to the next. In the ideal world, we'd collect multiple random samples in order to get a better estimate of the quantity we're interested in. In most cases, we don't have that luxury. Now, one thing we can do is look at how the variance of our mean values changes as our sample size increases, and hopefully choose a sample size that's going to produce mean values that are close to the overall population mean with a high probability. But all we can do is collect our random sample, and we know a lot about the probability distribution associated with the means associated with those random samples, and we use that to make inferences to our population. So some examples of random sampling, well, in the tea experiment that we talked about, if we randomly assign the treatments to the teacups as we're handing them to the woman that we're testing, that would obviously be an example of a random sample. Again, in remote sensing, verification of forest vegetation classification, we often divide our area up into a grid and then randomly select plots from that grid to go out and actually sample. Again, assuming that we aren't biasing it by only collecting those samples near a road, then each grid must have the equal probability of being selected in order for that sample to be truly random. There's different types of random sampling that you'll sometimes see referred to in the literature. You'll see random sampling with replacement. That's generally only used in very large populations. Maybe if you're sampling fish from a lake and you're catching them using some non-destructive technique and returning them to the lake, measuring length or whatever. In many cases, it may not be cost effective to actually mark those animals and when you put them back. And so you're acknowledging that there's a possibility that you could sample the same individual twice, even though it might be a fairly small possibility. That actually doesn't bias our estimates of the mean and the variance in any way. But generally, with the smaller populations that we deal with, finite populations, we often do what we call sampling without replacement. So we choose plots on our national forest with the stipulation that no particular location can be selected twice. Or if we're sampling individuals in our population of birds, for instance, we may capture individuals, put bands on them, measure their wing length, let them go, and maybe the next time we catch them, we don't collect that measurement because we only want it on different individuals in the population. And that's generally the way that we see this done. And again, it's an unbiased method of obtaining our estimates, and the variance is known. So I want to make a distinction between the sampled population and the target population. Target population is also sometimes referred to just as the population or the data frame. For instance, in remote sensing, we use satellite imagery to classify vegetation over large regions, sometimes entire continents. And to help inform the satellite data and the computer algorithm that we use for taking those satellite readings and translating it into different types of vegetation, grassland, shrubland, ponderosa pine forest, etc. We often have to go out into the field, choose locations, and compare the vegetation on those locations to our unclassified map, and that helps a computer determine the best ways to differentiate different vegetation types. Now, our target population, or our population, or sometimes called data frame, might be, say, the entire forest in Arapahoe National Forest. But when your boss looks at the amount of money that he's been given to go out and collect these vegetation data, he or she might decide, you know, we've only got $20,000 to go out there and collect 100 samples. If I send people all over the place out in the wilderness, it's going to take weeks or months to do that, and we simply don't have enough funds. So I'm going to limit the areas that I will go out and do the vegetation sampling to those sites that are less than 2 miles from a road. So it doesn't take people too much time to get out and back. There might be safety issues associated with sending them out in larger distances and so on. Now something fundamental has changed. The sampled population differs from the target population, or again sometimes referred to as the data frame. And we often have to grapple with this in environmental studies, simply because it may be logistically too difficult, maybe too time consuming, too expensive to get to every possible site within our target population. But we have to be careful about that and make sure we know what the ramifications are of that and how that may influence the results that we obtain. So before I go on and talk about the kinds of sampling you should be doing, let's talk about some of the approaches that occasionally we see used in environmental studies that can lead to problems. The first is what's called judgment sampling, where the investigator picks sampling units they consider to be representative. We sometimes see this with vegetation sampling. Plant ecologist says, this stand here is the most representative ponderosa pine stand in this national forest, so that's the one that we should use as our standard. This obviously will differ from the actual data frame or target population that we're making our inferences to, and that can often get us into trouble. So it's very, very important to know when judgment sampling is occurring and to be aware of the possible drawbacks in terms of really limiting our inferences from those kinds of data. Another type of non-random sampling is what's called accessibility, or sometimes called convenience sampling, where sampling units are selected only if they're readily accessible. For instance, the road issue that we just talked about. Sometimes this is unavoidable because of budget constraints, logistical constraints, safety considerations. But if you are forced into accessibility sampling, this should be made clear in the report or the published study that you produce. So at least the other people that look at this can be aware, oh, they only sampled within 2 miles of roads. And if you're studying black bears or mountain lions, which may avoid roads, then you know that the estimate of the density that's obtained may not apply to the rest of the national forest, for instance. What are some other issues associated with sampling near roads? Well, we know when engineers design roads and decide where they're going to place roads on the map, those sites tend to have less steep slope than, say, random areas on a forest or a region that you're looking at, right? They're not going to put a road across a steep cliff face or have it go straight up a steep mountainside. They're going to look for areas where the slope is less steep so that they can send their bulldozers up there easily, and also so that the cars can get up the road without too many problems. So there's some inherent differences between sites where roads are located and where roads are not located. And we also know that roads affect a lot of the things that we want to measure. Some species may be attracted to roads, other species are repelled from roads. So just being aware of that is very important. Third type of sampling we sometimes run into is what's called volunteer sampling, where sampling units are selected based on cooperation of the landowner. And a couple of things can start to creep in here. You can have what's known as non-response bias. So those individual landowners that don't let you go on your land or simply don't respond when you knock on the door and ask if you can cross their land, for instance, may be a non-random sample, again, of the area that you're trying to study. So if there's something different about those areas, if they're better irrigated, if they tend to be in valleys where there's more water, then again, you may be biasing the samples that you are obtaining. And in some cases, when a landowner does allow access to their land, they may force you to, cross or, do your work on some less desirable part of their land. So there can be a couple of ways that biases can creep in there. The fourth one I want to mention is what's sometimes referred to as haphazard sampling, where sampling units are selected based on some haphazard or unconscious criteria. It may seem like it's random, but it's not. And the important thing is, if you cannot determine a priori the probability of choosing any particular plot or site within the area that you're studying with a precise precision, you cannot make valid inferences from your sample to your population. So the fundamental issue with sampling is that all individuals or all sites within your study population are accessible and can be sampled, and you can determine what the probability of each one of those plots being sampled is. And we'll talk about this a little more as we get into the different types of sampling design. When you use non-random methods, it's, as we said, inappropriate to infer from the sample to the population, and there's also problems with determining what we refer to as the probability distribution of the mean of the sample, and that's often what we use for estimating variance and confidence intervals. So if we don't know what the distribution of those means are, then we often make errors when we calculate our confidence intervals. Now, whenever you go out and sample something, it's not enough simply to report the mean value that you obtain from your sample. You also need to include some measure of precision. There's various ways of doing this that you're probably all familiar with from your intro stats course. You can report the variance, the standard deviation, standard error, confidence interval, coefficient of variation, or CV, or the range. You see all of these sometimes reported. Probably the most common way of reporting information is with confidence intervals. And let's just talk a little bit about what we mean by a confidence interval. When we talk about a 95% confidence interval, we're saying that under repeated sampling, the trials, the true value of the parameter will be included within that 95% confidence interval 95% of the time. Now, when we go out and collect any individual sample, we don't know whether the true value is included within that confidence interval or not. If we were to go out and collect repeated samples, we would know that 95% of the time the true value is included within those confidence intervals. But it's a slight, subtle distinction, but an important one to make. The confidence intervals that we calculate are based on large sample theory and the normal distribution. We know that as you collect, take the mean of a sample, that distribution of that mean tends to converge on the normal distribution, and we can use that property from the central limit theorem to then make our confidence intervals. Now, these confidence intervals can be biased at small sample sizes and for values near zero. So in those cases, your confidence interval coverage may not be exactly 95%. Now, talk of precision and confidence intervals often brings up the issue of sample size. If you conduct a sample with too few samples, you're basically wasting your time and everybody else's time and money because you're not going to be able to obtain data with which you can actually statistically test your hypothesis. So, for example, we might want to know if logging impacts the home range size of red squirrels. So our hypothesis is that mean home range size of red squirrels is greater in logged or more highly fragmented areas. So you might go out and choose two similar randomly chosen forest areas with and without logging. Presumably you're trying to control for other factors that may differ among these sites. And you randomly catch N squirrels and estimate the home range of squirrels in the logged area, home range in the unlogged area. How big should your sample be so that you can actually come to a confident conclusion from your experiment? Well, we can look at this by starting with the two-sample t-test, which I'm sure most of you are familiar with from your intro stats class, where you ask, is there evidence for a difference, a statistically significant difference between two means? So that's the equation that we have here on the left, right? We have our two means that we're comparing. We have the variances associated with each one of those means, and we have some sample size that we use when we estimated these parameters. All we have to do is rearrange this equation to solve for n, and you can confirm that that gives you the equation on the right here. And this tells us right away the factors that influence sample size. So one thing we see in the numerator of this equation are the variances for the two samples that we collected. So as those variances increase for a given effect size that you're interested in, your sample size will have to increase. You also see that sample size increases as a square of the t value that you use, and that's determined by the alpha value that you choose. If you choose a smaller alpha, your t value will be larger, and therefore in order to test your hypothesis at that particular significance level, you're going to need a larger sample size. And then finally, in the denominator, you see the difference between the two means, actually the difference between the two means squared. So since that's in the denominator, as that difference gets smaller, in other words, if you want to detect a smaller difference, you're going to have a larger sample size. So you can pick the difference that you want to detect, and you can choose your t-value through the alpha value that you choose. But the variances of those two populations that you're sampling from are simply something that's inherent to the system that you're studying. And so in order to estimate sample size, you're always going to need some estimate of those variances, either through a pilot study or by going to the literature, looking and seeing what other people have found for variance and home range size of red squirrels, or you may have to make some educated guess. So to review this then, how do you determine the values of these various factors? Well, the alpha level, as we said, you choose, and that determines the t-value. The measure of precision, the variance in this case, is determined either from a pilot study, past research, literature, or a best guess. And finally, the effect size is based on maybe past studies that you've looked at and on something that you identify as biologically significant. In other words, you could collect hundreds of home ranges and be able to detect a change in red squirrel home range size of, say, 1/10 of a hectare. But the question is, does that really matter? Does that influence the biology of that species? If not, then maybe it's not something that you should be spending half of your lifetime trying to obtain. And this was just a simple example, of course, using the two-sample t-test, but this basic approach applies to all statistical tests, whether it's an ANOVA, regression, or whatever. And so estimation of sample size can be applied to all of those approaches, and there's often software available these days to help you with that. Okay, so let's go over some sampling designs. We're going to start with some simple ones, simple random sampling, and move up to some more sophisticated multi-stage approaches. I don't expect you to understand all of the ins and outs of these different approaches. That's really the purview of a different course, FW552 in our program. But I do want you to at least be somewhat familiar with the basics of these different approaches, because you'll often see them in various scientific studies, and it's helpful to have some understanding.
We already talked about simple random sampling, so I just bring it up here to indicate that this is our starting point. This is the simplest way to go out and sample our target population. Again, just to review, equal probability of each unit in that target population being included in our sample.
So let's talk about another commonly used type of sampling in environmental studies, and that's referred to as stratified sampling. In stratified sampling, what you do is randomly sample within strata. What are strata? It's groups of the population as defined by the investigator. So it may be habitat type, it might be soil type, it may be ecoregion. Whatever factors in the environment you feel may influence the variable that you're measuring. So again, if you're measuring the density of some bird or mammal species, you may have previous information that suggests that they're more abundant in one habitat type than another. So stratifying your study area by habitat would make sense in that case. If you're looking at the distribution or the size of different tree species, maybe soil type has an influence, and so taking that into account can be helpful. What are the benefits of stratification? Well, usually, and by usually, this really depends on whether you're good at selecting your strata, it results in increased precision. In some cases, you also may be interested in estimating your parameter, tree growth rate, whatever, by strata, and so you'll have that information that you can then use. And that sometimes will then obviously have influences on how you conduct your sampling. Now you can also allocate your samples to your strata differently. So you may, if you have two strata that are of equal area in your population, then you may decide to just sample each one of those equally. If you know that there's greater variation in the variable that you're measuring in one strata than another, then a more efficient approach is to increase your sampling effort in those strata where your variation is higher, and that will lead to a decrease in the overall estimate of the variance around the parameter across the whole population. You have to be careful with that, because if you choose incorrectly and oversample the strata that actually has a smaller variance, then the opposite will occur and you'll be less efficient in your sampling. But just so you know, you don't have to have equal effort by strata. So just for an example here, maybe you're interested in mule deer density on winter range, and you've divided the study area that you're looking at into two different habitat types, maybe mountain shrub in the green and ponderosa pine forest or juniper pine forest in the white. Okay, so you identify your two strata ahead of time, and in this case what we've decided to do is to place equal number of plots in each one of those habitat types. Now you don't have to do that. As I said, you could base your sampling scheme on the proportion of each of those strata in your study area. So if the green represents 30% of the total, then the green area would get 30% of your points, and the white 70%, it would get 70% of the points, and so on. Other examples of where you might stratify is if you're looking at fish abundance in some lake, you may stratify the lake by depth and sample areas of shallow depth in some specific way, and areas of greater depth in a different way. You may separate it by habitat if there's some vegetation in the water that fish may or may not be associated with, and you may want to stratify by that in order, again, to the overall goal here is to try to decrease the variance of your overall estimate. So just to finish up on stratified sampling, sometimes you'll see what's referred to as post-stratification of your sample after a sample has been collected. So you might stratify by sex or age after the study is complete. In some cases, you may not know what the proportion is of sex or age classes before you go out and sample. It may be difficult to actually target individuals of different sex or age class when you're obtaining your sample, so you just go ahead and collect individuals randomly, measure whatever you're measuring, and then in the aftermath you will stratify. Now there is a little bit of a cost, if you will, associated with this, because you don't know the exact number of individuals and what the number is in the actual population. You're just estimating it based on the sample that you collect. There's you add a little bit to your variance term to account for that, but generally it can be, again, a fairly helpful approach, especially if you know that there's big differences between your strata. So if you're interested in measuring the heights of men and women on campus, you may know that on average men are taller than women, and by stratifying by sex, when you estimate overall height of the population, you'll be able to increase the precision of that estimate by stratifying. You also can include auxiliary variables, and sometimes these are variables that you're interested in measurement or an experiment. So again, things like sex and age by collecting that information, now you can look at how the variable that you're interested in varies not only from males to females, but also has how it varies as males and females age in the population. Maybe that changes differently between the two. Sometimes you have inherent interest in those, like we did here. Sometimes you just want to use them to reduce the variation in the estimate that you're interested in. One thing to warn you against is just arbitrary stratification. So perhaps just dividing your study site into arbitrary elevation ranges from 3,000 to 3,500 feet, 3,500 to 4,000, for instance. Those may have no biological association on the ground. It would be much better in that case to identify habitat types, maybe that are associated with elevation, and use those to define your categories. But just using arbitrary strata often gets you into more trouble than it helps. The next type of sampling is what's called systematic or and cluster sampling. You might think, well, these things are totally different, but they actually share one fundamental similarity, and that is that the population is partitioned into primary and secondary units. And whenever a secondary unit of a primary unit is included, then all secondary units of that primary unit are included. This gets a little kind of convoluted, but basically with cluster sampling, you decide a priori that you're going to divide your units into clusters, and you'll go out and sample all of the units in that particular cluster when that cluster is selected. In the same way with systematic sampling, where you say have even spacing among your plots across a region. Once you determine the location of that first plot, then all of the secondary units are then determined by the location of that first one based on some distance between them. The important thing is that, again, you start with a random selection of clusters or of start points in your systematic sampling. So what are the benefits of this approach? Well, ease of use. In many cases, sampling clusters can be easier than logistically getting out to the same number of independent individual units. And as we saw with the random sample across our 20 by 20 grid, in some cases you will get a random sample which does not cover a particular region very well. If you use systematic sampling, you will ensure that you get uniform coverage across that entire area. Problems come up with this, though, related to periodicity. If organisms are distributed uniformly and your sampling unit consistently hits or misses certain elements, then it can get you into trouble. A good example of this would be like roads in agricultural areas. In general, roads are found exactly on section lines, right? And sections are 1 mile by 1 mile square. So if you're sampling vegetation across this region, for instance, and you happen to use a 1 mile distance between your sampling points in order to have a nice systematic sample that's spread across your region, if your first point happens to fall on a road, then every one of your subsequent points is also going to fall on a road. And that, of course, may not be at all representative of your total population. So just important to be aware of potential for periodicity in your population, and make sure if you're using systematic sampling that you're not using a distance that happens to coincide with that periodicity. So some examples, like we talked about, grid out a forest using GIS. randomly select a starting point within the first 10 cells of that grid, and then select every 5th cell after that. So again, you're getting into a nice systematic grid across the entire region, which can help with getting representation across the entire region, but with the caveat that be aware of potential for periodicity and that affecting your sample. For cluster sampling, maybe you're from a population of nests, you randomly select a nest and then select all nestlings within that nest to take measurements, for instance. So here's an example of a systematic grid pattern on the left, where you choose the first plot randomly using a random number table, and then each plot after that, the location is determined by the distance that you're sampling between those plots. On the left, what we've done is we select our center point randomly from our grid, and then we say, okay, and we're going to sample every single grid that adjoins that one. So you select this one, and then you sample across all of those. And here you can see logistically this may be easier if it's difficult to get to these locations. Once you're there, you might as well collect more information and get a larger sample size as a result. The downside, of course, is that those adjacent locations may have a certain degree of similarity to the center point that you've selected, and so therefore you have to account for the fact that they may not be what we call independent of one another. And should note, for systematic sampling, it's best to have at least 2 sets of primary units for proper variance estimation, because of some of the issues that we talked about of periodicity creeping into or being present in your population, which may then be repeated in your samples.
Well, as I talked about, systematic sampling is often an approach that's been used in large landscape studies in order to ensure that we have an even distribution of our samples across the study area. But I'm going to kind of jump to a newer approach that has been put forward and is starting to be used a lot. We're using it at the Bird Conservancy to select our 1 square kilometer grids where we do our bird samples across large regions. And this is what's known as generalized random tessellation sampling. Don't ask me where that came from. It's often referred to as GRITS sampling. And the nice thing about this is that it balances the advantages of systematic sampling with the unbiased estimates you get from simple random sampling. So it's a spatially balanced hierarchical design where the samples themselves are ordered in a linear fashion. And it's nice because it supports design-based, what we call design-based inference, so that you can take your estimates from each of your strata and roll it up to get an estimate of the entire region. And because the samples are ordered, what you often do is choose more samples than you think you're going to be able to get to, because sometimes some samples are inaccessible. Maybe they're on a cliff face, or it's on private property and the landowner won't let you get access. And what you can do then is just go to the next sample in your sequence and still maintain the spatially balanced random design. And just quickly, the way it works is you divide your area up into hierarchically nested suites. So you can see the first hierarchy is the large 0, 1, 2, 3. That would divide your area up into 4 quadrants. So you first pick randomly from that. Then within that, you randomly pick within the next 4 quadrats within that, and then do that at a third level to get the actual sample unit that you're sampling. Okay, and then when you choose your next sample or next sample unit, the grid sampling will ensure that it's not within the first level of the hierarchy in the last one that you just sampled. So in that way, it spreads it across spatially, still maintains the random approach of every cell having an equal chance of being sampled, and it's great for large landscape scale studies where you want to cover as much of the region as you can. You want to maintain that spatial balance across the region and be able to make inferences to the entire area. A lot of large scale programs are starting to use this now. Another approach that's fairly commonly used in ecological studies is what we call multi-stage sampling. And the basic idea here is that you divide your population up into samples, and then you randomly select those sample units and subsample within them. So in the example that we have here, say you have a large pasture that you want to sample, again for plant productivity or something, and it's going to be logistically difficult to get across that whole study area. So what you do is you first randomly choose, in this case we were 5 different sample units that we've randomly chosen for our sample, and each one of those is a 4x4 unit area. And then within each one of those sample units, we go and collect what we call subsamples. And in this case, we subsample 3 of those smaller units within each one. The advantage is that you go to that first sample unit, and it's pretty easy to collect all your subsamples because they're close to one another. And then you go to your next one, and again, they're fairly easy to get to. So this is often used, really the only justification for using multi-stage sampling is when cost is an issue and getting across your entire study area can be logistically difficult or very costly. So let me give you an example that I worked on recently for surveying bird harvest in villages in Alaska. We were covering basically most of the state of Alaska. We wanted to get estimates of total bird harvest in these rural Alaskan villages. And of course, it's very expensive to get to a village. And so once you get there, it makes sense to do multiple samples of households in order to get an estimate for that village. So what we did is we treated villages as our first stage in our sample and randomly chose, say, 20 villages in a region to go and sample, and then households were our second stage, and we take, say, 10 households in each village. Now, we're not going to every village. If we were doing, say, a simple random sample and we had 60 villages in our region, the simple random sample may force us to go to 40 or 50 different villages to get our sample, and in each one we might only collect information on 1 or 2 households. That's very expensive. So multi-stage sampling is used when cost is an issue. And if you're interested in it, I'd suggest taking a look at Thompson's 2002 book on sampling design.
Another approach is what's sometimes called double sampling or two-phase sampling. In some cases, it may be easier to collect information on auxiliary variables rather than the variable of interest. So a larger sample is taken and you're just recording information about the auxiliary variables. And then when you find individuals or sites that meet the criteria that you are interested in, then you focus in and collect the other information that you're interested in. So you have an initial sample where only the auxiliary information is obtained, and then from that larger sample you select a second sample, in a sense, that has the characteristics that you're interested in. Let's look at a couple of examples. So for instance, say you're interested in looking at the level of contaminants in lakes where common loons are found in the northeastern United States. Well, you might first do an aerial survey flying over many lakes in order to identify those lakes where loons occur. Maybe they only occur in 1 out of 10 lakes in the region that you're interested in. And rather than going to a random sample of lakes and spending a lot of time collecting measurements of contaminants at sites where maybe you have no interest, what you do is you do your first survey, identify all of those lakes where loons were observed, and then randomly choose from those lakes to go in and collect your water quality samples. Another example is maybe you're interested in whether students who don't exercise have more stress as measured by high blood pressure. Well, you might not want to just put out a general call to get students with high blood pressure, because they might have a number of factors that may lead to that. So what you might do first is just go out and do a survey and say, do you exercise or not? It might be pretty quick and easy to do that. And then from those students that do not exercise, you then take a random sample to obtain measures of blood pressure. Again, in these cases, you're really only interested in that portion of the population that has a characteristic of interest. So we're visually showing this here where maybe you went out and sampled all of these plots that are colored, but only the light blue ones had the characteristic species or soil type or whatever that you're interested in. And so those are the sites that you then go in and focus on and collect your additional, more intensive information. And there's what's called adaptive cluster sampling, where you initially draw a random or systematic sample, and if the object being sought is selected in a sample, then you sample the surrounding neighborhood until all objects are detected, or maybe you have some rule that you use of all adjacent plots or sites. This is especially good for rare objects, right? So if we're talking about snowy plovers, which tend to nest in colonies and are sparsely distributed across the landscape, then rather than going to a randomly selected number of sites where 95% of the time you're going to get a 0, you focus on those sites where you detect the species and then sample adjacent grid cells around that, knowing that you're likely to get positive values in those adjacent cells. So again, here's just a visual representation of this, where we have the animal of interest is present on these green cells. Our random sample are the dark red cells, and then sites where our random sample fell on top of a place where the species of interest was found. We then go to our adaptive cluster sampling, where we say, okay, we found the species here, so we're going to sample either all adjacent sites or keep moving until we identify cells where the species is not present. Then we're done with that cluster. We move on to the next and do the same thing. And again, this can be a very efficient approach where you have a rare species or a sparsely distributed species that is somewhat clumped where they do occur. And of course, you have to adjust for the fact that you're taking a non-random sample in this case, in order to estimate your variance. There are lots of different methods that can be used to estimate specific parameters from a population. Estimates are distance sampling to estimate the abundance of everything from marine mammals out on the ocean to birds in the forest. Removal sampling is an approach that's often used in estimating fish populations, where you go in with a seine, maybe catch a number of fish, count how many you catch, put them in a bucket, go and do it again. Presumably you're going to catch fewer fish because you've already removed some individuals from the population. You do that 3 or 4 times and you can come up with a pretty good estimate of the total population. All of these approaches still require some random or other appropriate sampling design underneath them. If you're interested in getting into more detail about sampling and sampling design, I'd recommend you take our FW552, Applied Sampling for Fish and Wildlife Studies, and it'll get into these methods in more detail. Whenever we go out and sample, we want to estimate some parameter of interest. And we do this with what we call estimators, or equations that take the data that we collect and produce the parameter we're interested in. That might be survival rate, population size, soil pH, whatever. So what are some good properties of estimators? Well, for one thing, we want estimators that are unbiased. So when we compute a mean from our sample, we want that mean on average to converge on the true mean. And secondly, we'd like to have estimators with minimum variance so that we can get the most precise estimate possible. Sometimes you'll find that these estimators have assumptions that underlie them, and you need to make sure those assumptions are met whenever you're coming up with an estimate. So examples of estimator assumptions with line transect sampling. The assumption is that all individuals in the population have the same probability of being detected. However, we know that maybe larger groups are more easily detected than individuals. Say if you're doing line transect sampling for ungulates across a grassland, for instance. You may miss individual animals that are bedded down, but if there's a herd of 10 or 20 individuals, you're probably going to detect them. That would be a violation of the underlying assumption. So you would need to do something to correct for that. There are ways of doing it, but you need to be aware of some of those assumptions in order to make those corrections. Lincoln-Peterson estimator is another common one that we often use. A lot of assumptions associated with that, assuming that you have a closed population, no movement in or out, no immigration, emigration, no tag loss, a variety of different assumptions associated with that. So again, just being aware of underlying assumptions associated with estimators is always important. And finally, just some examples of estimators. We all know about the mean, right? Where you take the actual observations that you've collected, those are your y sub i's, sum all those up, and then divide by little n, where little n is the total number of units in your sample. Nice unbiased estimator when your units are collected randomly. Linear regression analysis is also an example of an estimator. In this case, y is a function of the intercept, b sub 0, and the slope, b sub 1. Both of those are estimated from the data using estimating equations. And finally, again, the Lincoln-Peterson estimator is one that we use commonly to try to estimate population size. Interesting thing is that the simple formulation of the Lincoln-Peterson estimator is actually biased. Okay, so this is an example of a simple estimator where if you were to apply it without making any adjustments, your estimate of population size overall would be biased high. And we have, of course, N1 is the number marked and released on the first occasion, and 2 caught on the second occasion, M2, number of marked individuals caught on the second occasion. It's fairly simple to adjust this estimator to make it unbiased. You subtract 1 from N1, 1 from N2, and 1 from M2, and then subtract 1 from that overall product. But just want you to be aware that some of these estimators that we commonly think about actually have biases associated with them. So you want to make sure you're using the unbiased version of those estimates. All right, we are done with sampling.